Welcome to the Spotted Wing Drosophila webinar. For those of you who may not have done a webinar before, I'm Kathy Heidenreich, your host, and our speaker Greg Loeb will be joining us momentarily. Let me just give you a tour of our classroom here. If you look up on the bar at the top of the screen, you'll see a little person with their hand raised. If you hold down that bar, you'll see several options for you to make a comment while the presentation is going on. You can raise your hand, agree or disagree with a statement, ask the speaker to be louder or softer, to go faster or slower, show laughter or applause. Another way you can participate is down in the chat box at the bottom of the screen. You're able to type a comment or a question, hit the little conversation bubble, and it pops up. Just like that. I also want to point out some other features of the classroom to you. If you look over here on the left, you'll see there's a box with related links. The Cornell Fruit website link is there. The Pest Management Guidelines for Berry Crops link is there, and the Berry Webinar Archive. That Berry Webinar Archive is where this recorded webinar will be. So if you want to hear it again in the future, you'll be able to go to that link and hear it. It's going to be recorded. I see James and Jim have their hands raised. Do they have a question already? If you do, type it in the chat box. Good job. Okay, over on the right, you'll see that there's resource files for download, and there's a PDF. That's a PDF of the presentation that we're going to see shortly. And you're able to click on that, and it'll download to your computer, and you can print it. So if you want to take notes while you're listening, you'll be able to do that. Okay. Before we start, there's a couple of housekeeping things we'd like to do. We have a couple of questions we'd like to ask you. So we're going to have some polls show up on the screen, and we'd ask that you please respond to them. Some of those are for record-keeping purposes, and some are to get a feel for who our audience is and how much background you may have on spotted wing Drosophila. So here's the first one, very simple. Are you a resident of New York State? Everybody registered? Everybody voted? Okay, thank you very much. We'll close that one and go on to the next. Could you please indicate your occupation? And remember, you're not obligated to vote. But it's helpful for us if you tell us a little bit about yourself in this way. Okay, everyone had a chance to vote? All right, let's go on to the last one. How would you rate your knowledge of spotted wing Drosophila? Okay. Now the part you've all been waiting for. I want to introduce to you today Dr. Greg Loeb. He's professor of grape and small fruit entomology at Cornell University. And Greg received his PhD in entomology from the University of California, Davis in 1989 and began working at the Geneva campus of Cornell and at the New York State Agricultural Experiment Station in the Department of Entomology in 1995 as assistant professor. He has research and extension responsibilities for grape and small fruit crops, as I mentioned previously. Broadly speaking, his research expertise is in applied insect ecology, and he's particularly interested in plant-insect interactions, biological control, and integrated pest management. Greg?
Good afternoon, everyone, and thank you, Kathy, for getting this all set up. I know it's quite a bit of effort to kind of pull all the different things together to get one of these webinars to run seamlessly, and it looks like it's running pretty well right now. it was interesting to look at the poll, and it's kind of what I figured. I think most of you have some knowledge of spotted-winged Drosophila. Some of you have quite a bit, and probably more than I have in terms of experience working with this insect, and some of you really are just learning a bit about it. So the way I kind of oriented the talk was toward people that don't know a lot about it, and then I'm hoping through questions and comments we can kind of expand upon some of this basic information that I want to cover. So I don't have a lot of slides. I think there's 30 or so slides in this. So I don't think we're going to be rushed for time, so I really look forward to any questions you have, or if you can even have comments you want to add, if you can kind of put them in there in a succinct way in the chat box, that would be great. Kathy will hopefully keep reminding me if I don't see a question come through to act on it, because I figure as much as possible we can kind of deal with questions as we go through, and then at the end if we have more questions, that's great as well. Kathy, you can hear me okay?
Everything's great.
Okay, so I think I'll head on in then. Looks like there's a little bit of a chat going on in terms of not being able to hear. Do you want me to wait up a little bit, Kathy?
We'll try to work on that in the background.
All right, so I'm going to move on in. First thing I want to say is I really want to acknowledge a number of other people that are contributed both to this talk and our general knowledge of spotted-winged Drosophila. This is an invasive insect that wasn't originally, it didn't show up first in the Northeast, it showed up in other states. And so some of my colleagues in these other regions really have been doing the initial work, and I want to particularly thank Rufus Isaacs at Michigan State and Hannah Barak at North Carolina State for the work they've been doing on the sort of eastern United States, and then prior to that, lots of people have been working on it in the West for a couple of years. I also want to acknowledge that a lot of the photographs I have, I think all the photographs I have, are basically borrowing from other people, and I want to thank these people, and there might even be a few more that I've forgotten at this point to include here. Okay, so the outline I want to use today, oops, I went a little too far here. I want to cover sort of 5 areas. First off, I want to do a review fairly quickly of the history of spotted-winged Drosophila, a little bit about its basic biology, life history, and how to identify the adult stage anyhow, and just kind of go over that. Secondly, I want to talk about what I feel are the crops at most risk for spotted-winged Drosophila damage, particularly in the eastern United States and with a focus on the Northeast. Monitoring is going to be important for this pest during the invasion period and colonization period, and maybe important all the way throughout our dealings with it. So I'm going to spend a little bit of time talking about how we're monitoring for adults and a little bit for larvae. It's important to understand the phenology of this insect. I think it helps in terms of coming up with management plans. So I'll talk a little bit about what looks like the standard phenology for the timing of different life stages through the season. And finally, I'll finish up with some ideas on management. Okay, I'm going to head on then. Hopefully we're going to get some of the audio problems fixed as I go. Now, spotted-winged Drosophila is originally from the Asian area. It has a fairly wide distribution in Korea, China, Japan, in the Asian subcontinent. It first showed up in North America, in California, in 2008. And at that time, researchers asked, okay, what is the potential distribution of this pest based on where it's currently found in its indigenous area, based on its climate, temperature requirements, things like that. And they came up with this projection that highlighted the west coast of North America, up into Canada, and a big area of the eastern United States seemed to be suitable for this invasive species to become established. So that was sort of the prediction and projection in 2009, and that time it was really only known from the west coast, and maybe they had an occurrence in Florida. And right here is the current known distribution of this pest, and it actually looks an awful lot like, if I go back, and look at that projected distribution, the known distribution as of now is pretty similar to that. And it's probably still going to evolve, but it does indicate that the eastern United States is going to be an area of high colonization and establishment. So, as I said, it was originally found in California in 2008, affecting several fruit crops, and I'll talk about that in a second, up into the northern states, Washington and Oregon, and then into Canada. Showed up in Florida fairly early, but it's never really built up to much of a population in Florida, but has shown up in other states in the South. Michigan got it in 2010, as did North Carolina, where a lot of the research has gone on. In 2011, it really exploded up into the Pennsylvania and up into the Northeast, kind of showing up across that region, and sometimes at really quite high numbers. And everything looks like they're now established in the Northeast. It's not one of these sort of one-time establishments that may disappear. I think we're pretty much now in the same position as Michigan and out west, where it's established and something we're going to have to deal with. Okay, the life cycle of this fruit fly, and it is like other fruit flies. It has a fairly rapid development. That's a key important thing. We believe the fruit fly overwinters as an adult. Possibly the females overwinter better than the males. We don't know a ton yet about overwintering biology, especially in the colder climates. It's something we need to learn more about. But these adults can live 20 or 30 days, maybe longer in the spring as overwintered, mating and then laying eggs in susceptible fruit. These eggs are inserted into the fruit with the ovipositor of the female, and I'll show you a picture of that in a second. And then often at the exterior surface of the fruit, you'll see these sort of breathing tubes, white little fibers out of the exterior that kind of indicate an egg is sitting underneath that. After a relatively short time, these eggs hatch into maggots that feed on the fruit directly and develop over a 5 to 7 day period. Pupate either on the fruit, in the fruit, or dropping off of fruit, and they pupate in different places. They sit in that pupal stage for a number of days, then emerge or eclose and repeat the cycle. At the optimum temperature in that 70 Fahrenheit period, they can go through this entire cycle from the adult to an egg-laying adult in 12 days. So that's pretty quick. So there's a possibility of getting a number of generations during the season, and if things are surviving well, you have the possibility of building up quite large populations quickly. They seem to be somewhat limited by high heat in the summer. At least there's some indications that they can't tolerate very high heat. They may also be limited to some extent by cold, although they appear to be surviving in some pretty cold places based on our information in North America, and also based in work in Japan.
Greg, there's a comment from Richard Cowles that they've picked up one male in Connecticut about April 1st.
Okay, so Richard Coles has mentioned that they've had some trapping in Connecticut, and they've picked up a male in April. Farouk Zaman at Long Island, I was going to mention this a little bit later, but they've also been trapping through the winter, and they started picking up, I think they were collecting a few things in March and April, both males and females. So it's maybe unclear how well each of these sexes overwinter, but clearly they're surviving in these areas. I'll come back to this a little bit later, but they're already out and about, and presumably, if the adults are out, if they can find things to lay eggs into, they're going to be doing that. So one of the possibilities is that they're already starting their cycles. It will take longer in the cool weather, and the populations may not build up really quickly, and it will depend to some extent how successful the adults were, particularly adult females, overwintering. Now, one of the things I don't know, and it may be known, is whether these females are mated in the fall before they go into overwintering and are ready to lay right away, or they need to mate in the spring. Either way, it's probably a pretty quick process for them. Okay, identification, and you're probably pretty aware of this. The identification for purposes of monitoring are basically, based on the external morphology of the adults. And you can both identify the male and the female. The male is the easier sex to identify. As its name indicates, that the wing on the forehead and distal end of that wing has a distinct spot. And there are only maybe one other two species that have spots on the wings like that. I know one species we were catching, the males had double spots, and there's others perhaps out there that occasionally show up that have a spot. But if you find something with a spot like this towards the distal leading edge of that wing, it's likely to be a male spotted wing. In addition, the foreleg has these brushes on it that it uses, and they kind of appear as two dark bands, and you can look at that particularly under a scope or a good hand lens and see that on the males. So the males are relatively easy to identify. Females don't have those nice markers on the wings or legs. What they do have is a very robust ovipositor, and that's kind of shown on the right here, where they have these serrated scleroterized structures and that they use to cut an insert position in the fruit in order to lay their eggs. These things are quite a bit larger than most of our other Drosophilas, and they're a little bit hard to see using just a hand lens, but under the scope, and if you push on the rear end of the abdomen, you can get these ovipositors to extend, and it's quite remarkable. Once you've seen a few of them, you kind of notice how much bigger they are and more robust they are than other species. Practical purposes, males are much easier. You don't really need a microscope to identify it. Females, really helpful to bring it in, and it will take a little bit of practice to get used to identifying the females, but it can be done. But you really do really helpful to have a scope for that.
Greg, there's a question from Donald McLean. He's wondering what the first wild host fruit would be to watch in the spring.
That's a good question. I'm trying to think which ones would have I honestly don't off the top of my head know which ones might have fruit. I mean, something like I'm trying to think when the fruit of dogwood might show up, or another one is this what's it called? I'm forgetting. It's kind of olive in its name. One that I'll come to it in a little bit.
Oh, autumn olive.
Autumn olive. I'm not exactly sure. I know those things are flowering soon. I mean, autumn olive is probably flowering soon. Dogwood a little bit later. Hawthorn is maybe a potential. Some of those are going to be fruiting, but the fruit has to get ripe, and I honestly don't remember when those are ripening. I'm not sure how important those are, but I do think we need to look at it. It depends on sort of how high the populations are and how much they exploit those things. So even things like wild strawberry is obviously going to get ripe the earliest, but how much they actually get utilized by spotted wing, I don't know. That's the one that will be pretty early. But I think that's one of the questions we need to get a better handle on in terms of which wild fruits are being used, when are they being used, and how important they are in terms of populations.
Okay, there was a flurry of chat down there. So
yeah.
Kate
see a note. Yeah.
Is wondering about serviceberry. You talked about wild strawberry, and Eric is wondering about amelanchier. Richard Cowles commented they can also breed in flowers, and Laura is wondering if folks in Long Island and Connecticut are using vinegar traps.
Okay, let me go. So I don't know about serviceberry. That's a good question. We should look at it. The wild strawberry, I was thinking of Fragaria virginiana. I don't know about the other one in terms of potential fruit, but it's worth looking at a lot of these things. Now, Laura, I believe in Long Island they were using vinegar traps. I think that's true in Connecticut, but I might have to ask Rich about that as well. Rich Coles has made a comment that there's evidence that they can also breed using flowers, so that's interesting as well, taking advantage of the sweet areas in there. I see another question. This is great. Keep them coming. Can they use compost piles for breeding? Well, That's a good question. If the compost piles are not exceedingly hot, it's possible that flies could breed in there. Whether there was some rotten fruits and things like that they could utilize, I suspect it's possible, unless, if the composting isn't really done well in terms of getting that heat up. My guess is the heat would kill it. Farouk said and confirmed that indeed in Long Island they've been using apple cider vinegar for their kind of season-long or year-long sampling. Hopefully I addressed as best I could the questions, but some of these I just actually don't know the answer, particularly the wild hosts. I really hope to learn a lot more about potential wild hosts. Concurrently, work's going on in Michigan, in New Jersey, and I presume other states looking at wild hosts, because I think none of us, the researchers, believe this might play an important part in the phenology and population dynamics of this pest. Okay, I'll move on, at least temporarily. We can always come back to some of these questions and talk a little bit about what commercial agricultural crops are probably at greatest risk from spotted-winged Drosophila, and let's say the Northeast is what I'll focus in on. So I just have a table of potential hosts. And first thing is to look at the high-risk crops. For our areas, I think evidence is indicating that raspberries and the caneberries in general, but particularly the fall maturing, ripening raspberries are particularly vulnerable. Blackberries, late maturing blueberries, day-neutral strawberries. Again, when day neutrals, often we get fruit in the spring, and then again in the late summer and fall, and it's the late summer and fall. Strawberries that seem to be quite at risk. They seem to be quite highly preferred. Sweet cherries also are utilized. That's been a big problem on the West Coast. Whether it's as big a problem here on the East is still not clear, but it's certainly a potential risk there. That's kind of where they first really ran into problems with this, was in sweet cherries. But maybe that for us in the East, it's these fall raspberries that are going to be our most important one. In terms of moderate risk, a number of fruit crops are possibly to be exploited. One is grapes. We often get questions about, and I think we have a big question mark around grapes. Spotted-winged Drosophila can develop in grapes, will lay their eggs in grapes, but they don't seem to be highly preferred crop unless there's already some damage in there. And our observations, this really comes from Rich Cole's work in Connecticut. In 2011, grapes late in the season were actually hit pretty hard by spotted-winged Drosophila. Now, 2011 was a tough year for vinifera-type grapes that were ripening late in the season. We had bad weather, hurricanes, things like that, so that the fruit itself was in bad shape, and that may have exacerbated fruit fly damage in general, including fruit flies coming out including spotted-winged Drosophila. Some work out of California, some researchers out there have indicated that if the grapes are in good shape and intact, spotted-wing is not as likely to try to lay eggs in it, and it's not as preferred a host. East Coast situation, our weather is different, our conditions are likely to be different, so I think it's an open question on the risk of grapes, and I think that's something we're going to have to look at, as well as some of these other crops. We're just going to take some time to figure this out. Under moderate risk or even slight risk, I'm going to add now June-bearing strawberries. Now, I say this because for a number of years, when researchers, for the last couple of years anyhow, researchers have been looking in both June-bearing strawberries and day-neutral strawberries and have found no evidence of egg laying in June-bearing strawberries. They seem to be maturing before really the populations have built up to an extent that they're causing a problem, a noticeable problem. In 2012, in the Southeast, we now have reports of larvae being collected from June-bearing strawberries. Now, this is out of a researcher's field, was not being picked very clean, so there was a lot of ripe and overripe fruit in that field. and it wasn't at very high numbers. It was only a couple of maggots in a few fruits, but it suggests the potential is there and something we're going to want to look at over time to see if June-bearing strawberries are at risk for either economic damage or potentially simply acting as, at a low level, a reservoir for these overwintered spotted-winged Drosophila to get their generations going and building up, let's say, even on the residue of June-bearing strawberries, and providing the sort of the next step as they move towards the late season and build up to higher populations on things like blueberries and summer and fall raspberries.
Greg, there's a question from Jonathan.
Yep.
About how early they begin laying eggs.
I'm going to show you a little data here just in a second, but it looks like the fruit needs to be pretty ripe before they really start laying eggs into it, so that you're not likely much at risk with green fruit. And even fruit that's changing color and getting ripe, probably pretty low. It's really as they get ripe.
And Susan is asking a question about these compared to the regular household fruit fly. So
we have several species of fruit flies. A common one in fruit is the kind of fruit fly of genetics fame, Drosophila melanogaster. And it typically is only getting into fruit that is overripe and really almost rotten. And in fact, they utilize symbiotic yeasts and things that they actually feed on that really don't get going until the fruit's very, very ripe. So I think melanogaster can be a nuisance. It certainly shows up in our houses. We get them in wineries and can be a problem. But unless the fruit's being damaged by other things, those other fruit flies aren't as big a deal. This fruit fly is, I think, much, much greater concern because it's going into ripe, marketable fruit, so that there's a real chance, if the populations are high and you're not controlling them, of growers selling fruit with maggots in it, eggs and maggots inside, that look pretty normal, look really decent, so that they're willing to sell them. And that's the real risk from this pest, and why there's so much interest in figuring out ways of managing it and understanding its biology and managing it. So yeah, this thing's quite different, and it all comes down to that big ovipositor that they have that are able to get into intact fruit.
Bernie's wondering if early and mid-season blueberries are at risk.
I think over time they may become at risk. Right now the information suggests that they're not as likely, and it's the later season varieties where they're finding it potentially. Now, in blueberries, it's a little harder to tell in terms of commercial blueberries because they may be spraying for other things, like in our area anyhow, for blueberry maggot, and the same materials that they're using against blueberry maggot may be killing and likely killing spotted-winged Drosophila. So we'll need some more information on sort of unprotected blueberries and that kind of thing to sort of figure out which varieties are at most risk. Again, I'm suggesting the later season are probably the greatest risk, but over time, it's not inconceivable that as these flies show up earlier in the season, that other crops or other earlier maturing varieties are at risk. I'm going to look over the questions here because they're coming in, which is great. Hans has indicated, where to go, that he assumes thin-skinned grape varieties are more at risk, like Pinot Noir or many vinifera, than thicker-skinned hybrids and native grapes. That's probably true, Hans, but I'm not aware of any specific data that's tested and shown a big difference. I think most of the work I've seen has been mostly on vinifera, though, out of Oregon. But there may be some more information out there I'm not aware of that I need to look at. But it's a reasonable assumption, as well as a lot of our native hybrid grapes, especially our native grapes, are not going to be as affected by the fruit rots and things like that. So they're going to be in better shape and not have these injuries. Rich Coles noted, and something I meant to mention is that some of the things he's observed both in the lab and in the field is that color can be quite important in terms of attractiveness to these adult flies and for oviposition, so that dark colors, red colors, or dark colors, what they probably see, may be important, so that dark colored grapes may be more attractive to these flies than light colored grapes. So some of our red varieties, the Pinot Noirs, the Cabernets, the Merlots, and potentially the Concord grapes may be more susceptible. And it's again something that we'll need to look at. Yeah, okay. I get a question from Carol Enton. What about fruit sitting in open baskets at a farm stand? Would this attract ovipositing females, and customers could be going home with eggs and larvae and fruit? Sure, I would expect. It's very clear that these fruit flies use odors associated with ripe fruit. And if you had a basket sitting out there just like other fruit flies, you might get spotted wings attracted to those, and it's potential they could lay eggs. Whether those eggs would actually hatch, if they were taken home and put in the refrigerator and not or consumed right away, whether that would really be a huge issue in terms of the eggs. It would take some time for those eggs to hatch, and they would need to leave their fruit sitting out. So I'm not sure, whether a lot of that would come back to haunt you, but it's at least something to be somewhat aware of. And I don't know whether you might, it'd be something we want to look at. I know there's ways to put netting over these things. That would be a drag in a way, because you want your customers to be able to see the fruit and choose it. Yeah, we'd have to think about that as a potential risk. I guess initially I would say not a huge risk, at least in terms of getting maggots, possibly eggs. That's a good question, though. Let's see. Yes, I'm just confirming that. Yeah, the real danger in terms of marketing and economic damage is selling fruit with maggots in it. Okay. Wild hosts, I already could have talked about that, and I think we need to learn a lot more. I just list some of the ones that Rufus Isaacs and Michigan State has been looking at, and we're going to start looking at a number of these this year in close proximity to some of our small fruit crops. And that raised some interesting questions, that I think one of our other people were getting at. Could we possibly use one of these alternate wild hosts as a indicator plant to kind of help warn us of a potential risk from spotted ring egg laying in our cash crop? And that would be really quite helpful. I'll talk about monitoring more later, but one of the things we have found is that the traps, as we have them right now for catching adults, often only get adults after some egg laying has occurred in the crop. So it's kind of a delayed signal, and it would be nice to have a signal that says, okay, there's a lot of those fruit flies or some fruit flies in the area, and we need to really increase our vigilance at this time. So that's one of the reasons we're quite interested in the alternate hosts. I'm looking at a couple of questions here. Eric Sciori says, any skin damage will damage keeping quality, so oviposition is an economic concern in terms of rot during shipping. Oh, that's a good point, Eric. Thanks. So that's another aspect of the damage. So the puncturing of the skin could easily, that's a good point, if there's any kind of surface fungi, or actually we know these flies are probably carrying bacteria and fungus on their ovipositor, so that's a good point. In terms of you get some egg laying, that would shorten your shipping and storage, and that would be a drag. Kay Gilbert asked if they are attracted to dark colors, would they be attracted to something like traps using grape jelly? Yeah, that's a good possibility. They are attracted certainly to apple cider vinegar, and they're attracted to wine, so some fermented grape juice. They're attracted to bread dough that's started to ferment a little bit. This is work that Rich Coles discovered last year. So a lot of these aromas associated with fruit and even fermenting fruit seem to be attractive. Whether grape jelly would be the right thing. I mean, hopefully we're going to come up with other lures that are a little more compact and easy to use, but I think, again, that's going to be an active area of research in terms of what makes the best lure that can get these insects into traps for monitoring purposes, or hopefully down the road, something really attractive that we could use in an attract and kill station as a way to reduce the populations using an insecticide, but not having to apply that insecticide on the crop. Let's see. Elaine, do we know what spray to use when we should spray? Yeah, Elaine, I'm going to come to that towards the end of the talk. And there are some insecticides that actually are pretty effective against the adults. There's some issues related with that, but I will come to that at the end of the talk. Thanks. Carol Bragg asks, should vinegar traps be made with dark color containers? And again, I'll talk about that in a little bit in terms of monitoring, but it does appear perhaps that red-colored or dark-colored traps may be a little more attractive, although it hasn't been a huge difference between that and clear containers in terms of the traps. But it doesn't hurt, perhaps, to have dark colors. I think more important is making sure that you have a lot of places where the aromas are able to escape out of the trap and that the flies will be able to detect from a distance and come into. And I think that's a quite important trait. And also placing the trap in the correct position to optimize captures. And again, I'll say this again later, but coming up with something more attractive than apple cider vinegar would be nice. And there is work going on there, and I'm hopeful that we're going to come up with something that's actually a little bit better. Those are excellent traps. Now, I wanted to talk a little bit more about what makes a fruit more susceptible, and I wanted to show you a little bit of data from Jana Lee, who's an entomologist, works for the USDA ARS in Oregon, and she looked at blueberries at different ripeness and assessed egg laying and larvae in that fruit. So she looked at green fruit, green to pink, and then blue fruit. And you can see from this graph, let's see if I can find my pointer. I don't know where it went. Whoops, I turned it off. Okay, so you can see from this graph, in terms of eggs laid, not very many eggs in green fruit. She did get a little bit, but these are in kind of enclosures where the fly doesn't have a lot of choices, and so it would lay a few, but the vast majority of eggs were laid in ripening to ripe fruit in these blueberries. In terms of the ones that developed into larvae 2 weeks later, again, it was the ripest fruit that resulted in larvae. Hannah Barakin, her grad student, looked at this in a similar kind of question about this idea of ripening and thickness and toughness of the skin. And they looked at blackberries, blueberries, raspberries, and strawberries, different varieties that had different puncture strength, so different levels of toughness. So the higher this value, the tougher the skin of that fruit. And over on the x-axis, number of eggs laid in 20 grams of fruit. And as you can see, as toughness decreases, you get many more eggs for these different crops. With raspberry, being some of the thinnest, easiest punctured fruit, getting the most eggs. But that was similar to strawberries and blueberries and blackberries. So that again shows that it's probably skin toughness, skin thickness is a pretty important factor. And it also suggests the possibility, maybe Hans was getting at this with his question, that in terms of breeding for resistance, this might be one trait that can be exploited to reduce the egg laying. I mean, there's going to be trade-offs associated with toughness of the skin that'll have to come into play here, but that's at least one potential avenue that researchers and breeders can pursue. Okay, let's talk a lot more about monitoring, because I think of the situation that many of us are in, where the pest hasn't been here very long. We really don't know when they show up, where they show up, which fruits are greatest at risk, which farms are at greatest risk. We need a way to kind of assess the presence of these things. For right now, management of them, when you get them, is primarily based on insecticides. I mean, we want to find some other approaches, but insecticides are key. And key to that, and key to being able to continue to market your fruit, is detecting the presence of these flies and starting some sort of control program in response, some chemical controls program. So monitoring is quite important, we believe. I just got a quick look at a question here from Donald McLean. For quick trap counts, can one assume that if number of flies if the number of flies are caught and several are males, are other, let me read this more slowly, I'm not getting it all. All right, for quick trap counts, can one assume that if a number of flies are caught, several are males and are others likely females? Okay, yeah. So what's the relationship between the males that you can see when you just are looking with a hand lens or just your naked eye and number of females? Rufus Isaac at Michigan State did some work on this, and at least during the season, there's a pretty good 50-50 relationship. So if you're getting, 5 or 10 males on that trap, you're probably also getting 5 or 10 females. There is some suggestion that the females might show up a little bit earlier than the males at the onset of a colonization and infestation period, so that it's possible by only looking for males you might miss the initial start of that, but I think this is something we need a little bit more data on. Okay, so monitoring for spotted wing adults. And that's kind of where our focus has been mainly on. I mean, you could potentially also monitor larvae in the fruit, but what you want is an early detection before you get larvae, or as soon as you can, before you actually start getting infestation, to detect that the adult spotted wing is present. So researchers have been trying a number of different approaches. What seems to be a consensus approach that works pretty well is using a cup trap. It can vary in size. We've been using 32 fluid ounce deli cups made out of polypropylene, which are pretty strong, flexible material, last a long time. We've been using clear and being successful, although as I mentioned earlier, darker colors, red colors, may be a little bit better. We use 32 ounces. I think that's overkill. The lures that we've been using so far have been apple cider vinegar, a couple inches of that, plus a little bit of unscented soap to reduce the surface tension on the surface so you can collect the adults. At the top of the container, of course, we have a solid lid that keeps the rain out, but along the upper top of the container we have drilled holes, about 12 of these holes at 4 or 5 millimeter diameter around the top, that are used to both release the aromas of the lure, in this case the apple cider vinegar, and also let the flies get in there. I mean, you'll of course get other things in there besides spotted wing Drosophila. You'll get other species of Drosophila and other insects that can fit into that hole that it might be attracted to the aromas. So and these things will kind of come in here, maybe fly around a bit, and they get into the vinegar and die. We have a couple inches of vinegar, as I said, at the bottom of the trap. These traps should be hung as much as possible in the shade. When we're putting these in trees and in raspberries that have trellises, in vineyards with trellis wires, in blueberries on branches, what we've been using are these wire hangers that we get from pheromone traps, and shaping a sort of a circular area at one end and just putting the trap down, and so it holds the trap, and then attaching the other end of the wire to a branch or something. For something like strawberries, we're going to work with a stake of some sort and have some sort of strapping around that stake to put the trap closer to the canopy of the strawberries, near where the fruit is. I think that's a better place in terms of attracting the flies. I just saw a quick question from Susan Susan Selby asking whether apple cider vinegar is for color and can we add red food dye. The apple cider vinegar is really for the aroma that seems to be attractive to them, to these flies at a distance. The apple cider vinegar is pretty dark colored. It's probably dark enough. We haven't tried darkening it with a food add, some sort of dye, but that's an interesting idea that perhaps we'll have some other more insights on later in the season. I also see that Farouk mentioned earlier back when I was talking about sex ratio and how representative the males are to the females. He was saying during the middle of the season they were getting 50-50 males and females in their traps on Long Island. That's pretty much what we were seeing in our traps in the Finger Lakes, and I think Rufus was finding similar levels. Again, early in the season, the earliest in the season, it's possible there's a bias towards females. It's something we need to learn more about. What kind of soap? Bernie Smith was asking what kind of soap. I think any kind of not laundry detergent, dishwater detergent that's unscented is what we can use, and just need a drop of it in there. And in fact, I'm not even sure it's super necessary. That's just been general the recommendation. Oh, I get a note here from Richard Coles commenting that colored trap liquid can interfere with being able to see spots in the wings. Oh, that's a good point. So if you end up dyeing the fly, you might make it more difficult to identify. Thanks, Richard. That's a good point. Something that you certainly would want to look at as you were asking these questions. Okay, you've got these traps out there. Most people have been trying to visit them once a week. You filter the vinegar through some sort of filter system to collect all the insects that are in there, and we've been filtering the vinegar into an old apple vinegar jar to collect it so we can dispose of it elsewhere rather than just throwing it out nearby. And it's unclear whether if you threw out the vinegar nearby, whether it would become a source of attraction to the flies so they no longer be attracted to the trap. That's the reason to discard it away from where you're trying to monitor. Whether that's actually been tested or not, I'm not sure, but it seems like a reasonable precaution to take. So you would filter these flies. What we do anyhow is we use coffee filters that we in a coffee in some sort of sieve that we put the filter in. So we pour the apple cider vinegar through that, collect the residue. I've labeled that paper copy filter with the trap number. I generally bring these back to the lab and let them dry out for a little bit so they're not totally wet, and then scan the individuals for spotted-winged Drosophila. In my case, we have a scope so that we would look under the scope for their females to identify both males and females of spotted wing plus other species. And I want to emphasize that you're likely to catch other fruit fly species, so you need to go through that step of at least identifying the males. So you replace that vinegar weekly. Apparently, over time, the attractiveness of vinegar does decrease. So again, it's important to replace that lure regularly. And as mentioned, the last point here is that it's generally you start detecting these adults as fruit is ripening or is ripe. And I also, as I mentioned, the results from the west and in the east is it's not uncommon to not catch your first adult till after you've got evidence of egg laying. So that it's not an ideal trap for an early warning. It's just a point I made earlier, and we're very interested in developing more attractive lures. I am working with Peter Landolt, ARS scientist out in Washington, who's been doing some work on synthetic lures, and in fact, hopefully I and some other of us will be testing some of their synthetic lures this year to see if we can get an improvement over apple cider vinegar. Now, some of you might be familiar with work that's been done both in California and Michigan and Oregon on a different lure based on yeast and water and sugar. So basically you're creating this yeasty fermented mix and using that instead of apple cider vinegar. And there's some discrepancies in the results from the different areas. In Michigan, these yeast-based lures, although they're a mess to use, have been a little bit better in the beginning of the season, I believe, if I remember this correctly, at catching flies. This wasn't what Hannah Barak found in North Carolina, so it's sort of unclear how much benefit you get from using these sort of fermented yeast solutions as lures. I think we're going to need more work on that. Hopefully we'll come up with a synthetic lure that we can put in a rubber septa or something like that, or in a small polypropylene vial that we can use as an attractant rather than have to deal with all this, but we're not there yet. and more work is needed. But my point about this yeasty lure is that may provide some benefits, although it's a mess to work with in trying to sort out and get the flies out. I see a question here from Donald McLean mentioning that their experience in fall raspberries, that if they checked their traps on a daily basis, this resulted in increased catches, suggesting that, and I assume he means that they refresh their vinegar each day, kind of maybe emphasizing that the freshness of the vinegar is important. One of the things I don't know, and I'm not sure it's been done, is exactly kind of what Donald's talking about, is looking at a decay curve, like how attractive is 1 day, 2 days, 3 days, 4 days, 5 days, and looking at sort of what you're losing by waiting a week. Certainly for the widespread monitoring, it would be pretty difficult to change on a daily basis, but for a grower in a couple different sites, that's very possible. So that's an interesting point that it's worth looking at. Susan mentioned that, so if the traps, oh, sorry, let me read this. If the traps get out early, then if they're in the area before our fruit ripens, they are attracted to and caught in, are they still attracted to and caught in traps? So in other words, if we put these attractant lures out in raspberry before they have ripe fruit, are you likely to catch anything? I think the experience has been not really, maybe occasionally, but I think the experience has been that work better when they're near fruit that's ripening. I don't know, maybe other people have a comment on that, because I'm not really sure.
Greg, I'm wondering if you put your traps out, or this is Kathy, if you put your traps out before the fruit is ripe, are you actually drawing them to the planting? Are you shooting yourself in the foot, is what I'm wondering.
So in other words, bringing them in and then they just move over to your fruit.
Yes.
Given how rare it is to catch them before the fruit is ripe, I kind of doubt that, but it's conceivable. I would think, though, that we would be catching them in there before ripe fruit, and then they would maybe move over to ripe fruit. I know with strawberry sap beetle, we sort of see that if we put out bread dough in a strawberry field before we have ripe strawberries, we'll catch a lot of sap beetle. But as soon as the strawberries ripe, we stop catching them to nearly the extent nearly to the extent that we do before there's ripe strawberry fruit. And I think possibly the same principle would occur here for spotted wing. I wanted to highlight a comment that Richard Coles put through that I was going to mention at the end as well, is that it is conceivable and something that we want to look at in the future, and Richard's really taking the lead on this, is possibly trying to mass trap out the population of flies at a site. Let's say you're a raspberry grower. After the raspberries are gone, the flies are still around. Can you collect and kill by using these traps that are attractive and basically greatly reducing your overwintering population so you don't have as big a problem the next year? It's a hypothesis at this point, but something that might be possible. And if you can find the attractive lure, and even apple cider vinegar might work at that time of year, you might be able to have an effect. And that's something we would definitely want to look at. All right, here's a good question from Carol. How large an area can one trap cover? How closely should they be spaced? Again, we don't have real clear data on that. We're tending to put 1 or 2 traps in even as big as a 5-acre, probably 2 traps in a 5-acre planting. If you're only going to put a few traps out, I would recommend putting them out on the edge of your planting. There's some evidence that the flies are coming in from the edge, and you're more likely to catch them at the edge first. Whether you would benefit from having 10 traps out there, it's hard to know. So 2 to 4 per a 5-acre planting would probably be sufficient to detect their presence, though I don't have specific data on that. And how close they should be spaced? I'm using something like, I don't have specific data. You want to have them, I'm going with 10 to 20 meters apart for our traps. when we're putting multiple traps in a field, just so they're not kind of competing with each other. But again, we haven't done the experiment to kind of test what that optimal sphere of influence is for a particular lure. Carol asks, is trapping for monitoring and not, yeah, I'm sorry, I didn't see that. Yeah, so trapping is primarily at this point for monitoring, not for protecting the crop. So, you know, we've used mass attraction and kill for some other pests successfully in some conditions. Even for something like a blueberry maggot, in small plantings, we've been able to use attractive things at the periphery and be able to actually reduce the overall population. And is something like that possible for spotted-winged Drosophila? I think it's possible. You know, it depends on how far they disperse from other areas and things like that, and getting a really attractive lure put out at the right place. And that comes back to something Rich was suggesting, is an attract and kill kind of bait station. Donald asks, will spotted-winged Drosophila be moving over distances as last year, region to region? That is a good question. I am guessing that the populations are going to build up in the south like they did in the previous years in North Carolina, Georgia, up into Virginia, and in the right kind of climate situation, with the right kind of storm fronts coming up. Sure, some spotted-wing, maybe even a lot of spotted-wing, can be redistributed through these storm fronts. We don't have a lot of data on that, but it certainly makes sense, and it fits the pattern we saw last year after some of those big storms came through in the fall, and all of a sudden we had spotted wing everywhere. And I think that's a really good question. So, and I don't think we completely tested it. So last year, they just exploded and showed up everywhere, all the way up into Maine and beyond. And they seem to be corresponding with these storms to some extent. How well will they survive in these more northern populations? I don't know. In this winter, probably okay. If it was a normally cold winter, maybe not so much. So I think it's going to maybe vary and depend on the year how important the dispersal is from the south versus the population growth from the populations that overwinter. As a good question, and one I guess we really don't know the answer to, but I wouldn't be surprised if long-distance movement from the south is not part of the dynamic for this pest, much the same way that it is for something like potato leafhopper. And we may find that certain regions are more vulnerable to where these storms drop them off than others. That would be something that we want to get a handle on. Okay, and the question is, do we know how far adults travel? And there's sort of two types of travel they might be doing. Something that the entomologists call trivial dispersal, and that's sort of local movement, you know, from this raspberry field over to this other raspberry field or to the farm next door. And we don't have a lot of information yet on their sort of daily movement. They clearly can move, you know, meters, tens of meters. Are they moving kilometers? I doubt it. Certainly not on a daily basis. There is work going on in California and Oregon using mark, release, recapture techniques, or mark and recapture techniques to try to start getting an estimate of this. For example, looking at the distance movement from fruit flies that are coming from wild blackberry, in this case in the study in Oregon that I'm thinking about. They're marking those flies as they're emerging out of that wild crop where they think it's an important source, and then looking for these marked flies in different areas at different distances. So we will be able to start getting a handle on what we are calling sort of local or trivial dispersal. Now there's long distance dispersal, the type that I think we did see last year on these storm fronts, and those can be quite long, obviously. How often that occurs and how many of them travel that way, we don't know, and something we're going to perhaps want to get a better handle on. But from sort of on a farm basis, we don't yet know. I think they're mobile, but how mobile, I'm not sure. I get a question here. What areas of New York have they been seen in? From Elaine, and I'll come to that in a second. But in eastern parts of New York in central New York is where we've seen the most of it, and I'll show you some data on that in a little bit. Bernie Smith asks, so on a farm with a lot of woods and a lot of wild grapes, etc., are you saying traps will help? I guess what I'm thinking about in terms of wild hosts near a farm, that traps in those wild hosts, that they may be an early warning system that the crop plants are at risk. Not necessarily that traps would help kind of eliminate them like a mass trapping, but that might be a place where you might be able to use, and that the adults would be captured sooner than they would in, let's say, your raspberries, your fall raspberries, and you could use that as a early warning that, okay, I need to be doing something with my raspberries. That is another question. Well, let me finish up on this, Kathy. Related to that, one question you might ask is, well, if these wild hosts are really important, or a certain couple of species are really important and the flies don't travel very far, does it make sense to try to remove some of these wild hosts? And that's a good question. I'm a little bit doubtful of that, but it's something we need to think about. Especially we could find that certain wild hosts are particularly attractive to the flies, and there's not so many of them that it's not impossible to basically remove, and that the flies aren't moving so far that your effort right next to your farm is being overwhelmed by things you can't control. Hopefully that made sense. Kathy?
Yes, I just wanted to comment, Greg, that we're already at 2 o'clock, and if we could just hold some of the questions so that we can get through the rest of the presentation, there'll be time for discussion at the end.
Okay.
So feel free to put your questions in the chat box, but let's continue through the presentation and we'll get to them at the end.
All right. Fair enough. My last question was Bernie's on wild host plants around the wood edges. I want to mention that it is possible to look for the larvae or even the egg, that presence of eggs on the surface of the fruit with a hand lens. And for small plantings, or well, not just small plantings, but you may be able to get a sense of it. For some fruit, it's not very obvious that there's egg laying going on unless you look pretty closely. So you can see here some pictures that I'm not sure, actually, maybe Rufus sent me with some of these on blueberry, both the puncture holes and then the breathing tubes. With a hand lens and a good light, you can see this. You can also look in the fruit for the larvae. We can use something called a flotation technique, where you take a collection of ripe and overripe fruit, maybe mash it up a little bit, and then pour salt water on it at that ratio of 1 tablespoon of salt per 1 cup of water. And in that condition, the larvae tend to float up to the top. Now, you can't tell if that's a spotted wing Drosophila larva or another species of fruit fly, but it can at least tell you that you've got some infestation going on here. And that can be useful for some crops, let's say for fall raspberry. If you detect some larvae, okay, you don't want to sell what's currently on the planting, you can do a real clean picking, put in your control measures, and then anything ripening beyond that should be well protected. So that can be helpful technique. And we'll be doing some of this in conjunction with rearing out flies this summer. Okay, a brief mention of phenology. I've kind of alluded to this a number of times. Like, when do these flies really build up populations during a season? What's their seasonal dynamics? This results from a unmanaged blueberry field in Michigan, provides what the general pattern has been for the bulk of the population. So this is a field Rufus was monitoring with his traps, using apple cider vinegar in these traps, and they monitored throughout the season, and maybe they caught their first adult in early July, but populations really didn't start taking off until late August, into September, and into October. So presumably those fruits that are ripening at that time, particularly fall raspberries, late season blueberries, potentially grapes if they're vulnerable, are the ones, and day neutral strawberries would be out here as well. Those are the ones that we think are going to be the most at risk because of this general pattern that we see of populations really only building up to huge numbers late in the season. And this is something we're seeing in the east, and they also see it in the west. When they show up is a little bit earlier In the more mild climates or where the flies have been around longer, I think Rufus is starting to see flies, initial catches a little bit earlier. I think Hannah in North Carolina is seeing them a little earlier each year. So we may see some of that, but really the big populations are building up late in the season. And hence why the fruits that I've identified, I think those are the ones we're going to be at most at risk and in practice have been. Now, real briefly, for New York, I wanted to mention what we did last year. A number of us initiated monitoring spotted wing during the season last year. Farouk Zaman and Dan Gilrain started putting traps out in September in Long Island of 2011. Peter and Laura were looking for them in later season in raspberries in Hudson Valley. I think Farouk and Dan were looking at a number of different crops. In the Finger Lakes, we were looking at a number of different crops, and we actually monitored using these adult traps throughout the season, and Tim Weigel was looking in grapes. Our first collection, this is the first capture known in New York. They may have been here earlier, but the first one we got was in the end of August. Peter found this in Hudson Valley, I believe actually in a vineyard. Remember, these are adults, though. Most of our captures, though, were in September, October, and then onward. And in fact, in Long Island, Farouk, I didn't have all this data to show. Farouk got some incredible numbers of traps late season on Long Island. So they were showing up in Hudson Valley, Long Island, Eastern New York in general, but also the Finger Lakes. Tim Weigel did not get any detections in Western New York, but he mainly only sampled in vineyards. There was some detections in northwest Pennsylvania. Actually, excuse me. Yeah, the north central west Pennsylvania. So they're in that area as well. The crops that they showed up in, I got to remember all these traps were for adults, and that may or may not tell you whether the fruit were infested. But fall raspberries, both our observations and observations coming in from some growers in the region, indicate fall raspberries were particularly the ones hit by spotted wing in 2011, with some growers virtually having to cease harvest and sort of close the plants down for the season because they just had too much infestation and were not able to get on top of it, or really didn't want to have to resort to two or more applications of insecticide to keep them clean. I wanted to show you a little data real quickly from Long Island up into October. Farouk had traps, these apple cider vinegar traps, in a number of crops, peach, apple, tomato, raspberry, and grapes. So these are for adults. And again, he had his traps out in early mid-September, didn't catch too much. Then in October, really started catching a lot. And he caught, that time, the most in this raspberry field, where I think he was also seeing evidence of damage in the fruit. He also caught a lot in this apple field. Where they were coming from, it's unclear. This was not well harvested. I think there was apples sort of falling to the ground, so it's possible the apples that were breaking down that were producing a lot of flies, or for some other reason they were congregating in this apple orchard. We don't think apples are a crop at risk, however. He did also want to point out grapes. They have a lot of vineyards out in Long Island. He had traps out there. Wasn't finding much evidence until quite late. But they're not necessarily harvesting all their red varieties until even into late October, November. And Farouk can correct me, but I believe he had some evidence that the fruit was being infested at that late season with some fruit flies and some spotted wing. I don't think he directly reared them out, but he had some evidence for that. And as I said, Rich Coles found similar things in Connecticut. To illustrate the fact, this is again data from Long Island. This is the percentage of spotted wing Drosophila over time, and basically the point here in these traps is initially the spotted wing was the minority species of Drosophila, but by the end of the season or late season, it became the dominant species. The other fruit flies are dropping out of the system, and I think that's a common pattern. And I guess the real question, and Farouk kept monitoring and had even higher peaks later in December, or kind of astronomical. I'm sure they got some freezes, but after the freezes they would still catch a few adults, and he has been catching pretty much adults most months in February or March anyhow, and in April at low levels. So the adults are overwintering there, and as we mentioned, Rich mentioned that they're catching some stuff already in April in Connecticut. So they're overwintering. Where they're overwintering, that's a good question. We need to learn more about that, what their survival rate's important, and sort of how they get going in that season. I think all important questions. In central New York, real quickly, we also use these vinegar traps. Now, we had the traps out all season. So the data from Long Island and the Hudson Valley, those traps only went out in September. So, okay, maybe the fruit flies were present earlier in the season. And we had traps out for the whole season using the vinegar traps in raspberries, cherries, apples, grapes, and blueberries. And I had an intern working on this project, bless her heart, and she changed these traps every week through the whole season. Never found one spotted-winged Drosophila. Pretty discouraging. A couple weeks after she went back to college, we started finding them, particularly close by raspberry, but also in some other areas. So end of September and into October, November, then we stopped trapping. We caught significant numbers of both males and females in the Finger Lakes area. We had traps up further north into Wayne County, Lake Ontario, but as we got later in the season, we sort of stopped those traps. Up till mid-September, we hadn't caught anything. My guess is that some did show up later in the season, like happened in a lot of other areas in the Northeast. Okay, I want to talk a little bit about management. And I'll go through this fairly quickly. I guess we're kind of running out of time. Cultural. I'll go through cultural, biological, and chemical. So good sanitation is probably quite important in a commercial field where you're regularly picking, let's say, raspberries and blueberries, day-neutral strawberries. So keeping the level of infested, or excuse me, of overripe and ripe fruit in the field down to a low level. That's likely to help in combination with other control measures if you have an infestation. As I mentioned earlier, maybe there's some benefit, with a big question mark on it, of removing non-crop host plants or wild hosts in the close vicinity of the field. There's also some evidence that we need to look at more carefully, that if you're picking fruit for market, can you eliminate and kill off any eggs or larvae, well, let's say eggs, by cold storage? And there's some evidence that there's some susceptibility to refrigeration, and we need a lot more information on that. But that might be something for, even if you're picking and doing roadside stands, but for retail, if you could utilize cold storage for, let's say, a 24 or 48 hours to kind of eliminate the presence of these insects, that would be quite helpful. Biological control. What you would hope is that as this invasive insect is here longer, that some of the native natural enemies, the parasitoids in particular, would move from other species of fruit flies onto spotted-winged Drosophila. In Michigan and in North Carolina, where they've been looking at this for a couple of years anyhow, they have yet to detect any parasitism of larvae of spotted wing by parasitoids. However, in Oregon, Peter Scheer has reared out 2 species of wasps from the pupae of spotted-winged Drosophila. These, I think one of them is in the family Pteramalidae. I'm not sure the other one. I don't know how prevalent they are, how important. Probably they tend to be kind of hitting the late instars of this species and not at particularly high numbers. So perhaps these species are not going to be the important ones, but maybe others will be. There's also active interest in going to the area of native origin of spotted-winged Drosophila and looking for natural enemies that are doing a better job of regulating their populations in Japan and other parts of Asia, and bringing those natural enemies back to the United States, making sure they're not going to be at risk for other species in the environment, and then releasing those as a way to control it. So I guess long term, one really hopes that we see some more activity on the biological control stage, partly because of the importance of all these wild hosts. We're never going to be able to get out to those and exert chemical control on it, so we really need some biological control to help us out here. So chemical control. In the short term, this is probably our main approach to managing spotted-winged Drosophila. Fortunately, a number of insecticides are fairly effective targeting the adults. This is a graph of some data that Rufus collected where they treated blueberry with different insecticides, then exposed those things to adult spotted-winged Drosophila and assessed larvae 1 week later. And they looked at a number of compounds that we have labeled in fruit crops. Delegate and Entrust are spinosad type products, imidaz, an organophosphate, lanates, a carbamate, Malthone is an OP, Mustang Max is a pyrethroid, and Paganic is an organic natural pyrethroid that can be used. So of these, the spinosades have been working quite well, as well as the organophosphates. The pyrethroids also have been generally effective, so that's good. We have a broad range of insecticides, some of them fairly narrow, low-risk materials like the spinosades that are effective. Paganic has not proven particularly effective for organic growers. That's one of the things they can resort to. Entrust is another one for most of our crops, our small fruit crops, and that's been more effective. Although you worry that overuse of these things is going to result in some resistance issues. Summarizing the work of Rufus and some others, as I mentioned, some of the broad spectrum insecticides, the OPs and the pyrethroids in particular are effective. The neonicotinoids have not been particularly effective. The spinosins, Delegate in particular, and Entrust have been effective materials. For New York, now in New York we have this requirement that the pest needs to be on the label if you're going to apply for it. So we have need for 2EE label exemptions or expansions. So in New York we have a little bit more limited materials available at the moment, so that we are kind of restricted to Delegate and Entrust in caneberries and bushberries and strawberries. Now, for strawberries, for Entrust, actually, we don't have that label. That's kind of a misprint there. There's a 2EE for Entrust, but it didn't include strawberries on that. I got to check on that, actually. In strawberries, we do have Radiant, which is the same thing as Delegate. That's effective. My sense is that other insecticides, well, I'm certain other insecticides will get labeled with 2EE label exemptions or expansions, so that I'm sure we'll get a pyrethroid and a carbamate that we'll be able to use in New York. One of the key things, though, for all these products is we need a very short days to harvest for these chemical applications, which we need to be putting on at harvest or near harvest. So we're going to need to take that into consideration, where we only can really afford 1 or 0 or maybe 2 days to harvest in order to use them. The upshot here is that I think New York and other areas that are IPM programs in small fruit crops in particular, which have not been particularly insecticide intensive in the past, are going to be disrupted to the extent that this spotted-winged Drosophila expands and becomes established in our area. And we're really going to have to work hard at accommodating this problem and looking for alternatives. I do believe monitoring right now, sort of in the year and the next years to come, is going to be important. Not everybody's going to get them. They're not going to necessarily get them at a time that's a problem. So again, I want to emphasize the importance of monitoring. I'd hate to see people just go out there and start spraying twice a week without really knowing they've got a problem. And that really brings up another question we really want to get some information on is, what is the frequency of sprays necessary? How do you decide that? As well as we need to start looking for alternatives. Speaking of alternatives, I wanted to say that there is some interest going on. I've mentioned several of these, and I just heard today from Rich Coles that a grant that he was the lead on to Northeast IPM has just been funded to begin looking at some of these alternatives. And I guess in the medium term, I have some real optimism that we're going to come up with some alternatives. So a couple of things that we want to look at are this idea of attract and kill stations, and we've got a number of ideas for things that are attractive, and looking at when we might put those out. we've been interested in looking at this idea of pushing and pulling using repellents in the crop, and then putting an attractant potentially with an insecticide associated with it on the outside of the crop, something that has some potential you want to look at for some crops. Richard's done some work, for example, with kaolin clay and some other materials, but kaolin clay seems to be quite a repellent, and for some crops such as grapes, we may be able to use that as a push and then have something, whether it's a vinegar or one of these other attractants on the outside of a vineyard, as a place to get the flies to congregate and perhaps to kill them and keep them out of the vineyards. So I think we're going to see a lot of work, hopefully in the next few years, on these alternatives, so that we're not really only relying on chemical control. Biological control, I mentioned the work going on in terms of international exploration for parasitoids that potentially could be released, but this is a very long-term prospect. It takes a number of years to identify candidates and then to show that they're safe to be released. I want to mention Rich is doing, I think, some pretty neat things, and he wants to work with entomophagic fungi and bacteria, Bacillus thuringiensis types bacteria, and doing some actual breeding, selecting experiments where he's going to look for highly virulent fungi or bacteria that might be used against spotted ring Drosophila in some manner that we could take advantage of. On that grant with Rich, we also wanted to look at some ways to increase the effectiveness of insecticides using phagostimulants. I didn't list this here, but Rich has shown that, like a lot of other flies, they're stimulated by sugar and some other things to feed, and that maybe we can combine sugar with a toxicant and spray it on fields and utilize that to enhance the efficacy of these insecticides and perhaps drive these populations down to really low levels. So there's things coming that I hope that will be helpful. In addition, a number of us are on a large, on a grant from Northeast IPM to form a spotted wing Drosophila working group. This was headed up by myself, Julie Cesar Rodriguez-Siona at Rutgers, and Dean Polk at Rutgers. But a lot of people are going to be involved with this, and our objectives here are to work in the Northeast to identify and prioritize research and extension needs, develop effective lines of communication among various groups, including the growers, and facilitate sort of a networking with other people, and also facilitate development of real guidelines that we can utilize here. We will be having a winter meeting to develop these priorities that many people will be invited to sometime, I'm guessing now in October, and we haven't got that date figured out, but we're going to do that soon. So look forward to that, and I think that'll help us. One other major project we hope to see coming is a project funded by the USDA Specialty Crops Program. This was headed up by Hannah Baraka at North Carolina State, but involves a number of us in the area. This would be significant research to develop a better understanding of the biology of this pest and to develop optimal control methods. You can see the objectives there. One of the things we're very interested in are the landscape and production risks, which landscapes and systems are most at risk for spotted ring Drosophila, perhaps being able to develop risk management protocols. The monitoring, as I mentioned throughout this talk, is an important one, and Cesar at Rutgers, Rodriguez Solano is going to be working on that and doing a lot to help develop that, and we'll be working with him. We also will be working with chemical control and best pest management strategies. So we should be hearing about the fate of that grant. It's a several $million, I think $3 million proposal, probably in the next month. So hopefully that will come in. If it doesn't, we'll probably resubmit for next year. We really believe that the Eastern United States is a critical area and unique to Western United States, and that we deserve and warrant support from the federal government. Finally, there are some number of very good websites. I don't know if I'll have time to go through all these, and I think many of you are aware of them. I believe the website that Rufus and his colleagues have put together at Michigan State is quite good and worth checking out. There's even a video there that Rufus has done on monitoring that I think is worth looking at. Oregon State was one of the first to get a website going, and they have a lot of contemporary information, current information coming up. Hannah Barak has got a website including a site that's mapping out the discoveries of spotted wing adults in traps, usually a trapping system where people are reporting on that. I'm going to click on this real quickly. You can do that as well, I guess. If you go click to that, it basically comes up with a map, and that map, as it will load up, will show for 2012 and previous where they're finding spotted wing so far in the mid-Atlantic, southern, southeast areas. At the moment, we don't include northeast regions, but there is plans to utilize Hannah's site to expand it up into the northeast so that we could be reporting on this as well, especially in the early parts of invasions, early in the season and during colonization. It might be quite helpful to have a network of stations reporting on when it's being shown up in different areas, so that if you're a grower in an area or an extension educator in an area, you can be kind of paying attention to that and helping you be alert to potential risk that's coming in. I'm going to go back. One other site that it's not really worth going to now, but when we meet and develop our priorities and get information together through the Northeast IPM Center, we will have a website as part of their working group websites and getting our information, and that's where we'll post it. If that spotted wing, or excuse me, that SCRI grant, the specialty crop grant, comes in with Hannah, one of the goals there will be to coordinate our extension efforts throughout the Eastern United States. I don't know if you've noticed the symbol I've been having on my slides. This is the symbol we're using with this group that we're calling an E-fly, and it's a sort of a symbolic of our desire to coordinate our efforts in the Eastern United States so that we're sharing information, sharing tools, and sort of making a coherent effort at dealing with this problem. All right, I apologize for going long. I really appreciate the questions. We got a lot of more questions that we can to work on, but that's kind of the end of the slides. It took me a little longer than I wanted to go through, but I just wanted to thank a number of people that have contributed in various ways to pulling this together, and I guess I'll let Kathy direct us on to the next steps.
That's the end of our formal presentation, but we still have bunches of questions. Thanks all who are here. You're welcome to stay and we'll go through those, and if you need to depart, feel free to do that as well. Greg, we left off with Kay Gilbert. She wanted you to give her some idea what storm fronts are, what we were referring to, so she could be on the lookout for those.
Well, yeah, I'm not sure if I'm the expert on storm fronts, but certainly things like these nor'easters that come up where you get this low pressure system forming along the southern, southeast or mid-Atlantic areas, and they're bringing a lot of south wind, and those south winds would be carrying all sorts of organisms up north, and we see this for a number of insects and diseases for that matter that can get transported on these quote-unquote storm fronts. So they're low pressure systems, I think, that tend to come up from the south.
Donald is asking about trap action thresholds, and then he wants to know how does treatment schedule interact with SWD growth and development.
We do not have explicitly a trap action threshold in the sense that, if you catch fewer than 5 adults, don't worry. I mean, I think if we have anything, it's if you catch any, then at this stage, especially for a vulnerable crop, if you're catching it and you are growing fall raspberries, that says you should be probably doing some chemical control. I mean, I think that's the way we're going to have to look at it for the time being until we come up with some other better information. But if you've got it, just like we say for blueberry maggot, if you get trap catches of those things, then you initiate almost a calendar-based spray regime until you're done harvesting. I do believe that's probably what we're going to have to do for these vulnerable crops. Does treatment thresholds, how does it schedule interact with spotted wing growth development? I'm not sure I understand the question. Kathy, do you have some insights there?
I don't, and maybe Donald will give us some direction with that in the chat box, and we'll move on to Jonathan. Is the pattern established in areas where the population has been established? I was worried that they arrived sometime midsummer and weren't noticed until they build into high numbers in mid-August.
Well, it's likely that, and I think Connecticut would confirm this, that they were picking up some spotted wing prior to these storms that I mentioned coming up. So at least in Connecticut, they were probably present at low numbers. I don't know whether they were present in, Vermont, New Hampshire, up in upstate New York, let's say in July. They could have been, although we didn't catch any in our area. So it's probably a combination of both in some regions, but the bulk, I'm guessing, for this last year, 2011, did come up and really didn't get going until late season in terms of these high populations. Maybe a different story in 2012.
His comment is that it would be good to survey the group, and I'll put those polls together if you want to go ahead with Carol's question.
Yeah, to ask in terms of folks, particularly the growers.
I'll put together a poll for that if you'll go on with Carol's question.
Okay. Any suggestions for how to rear out larvae? Leave on plant with a net bag? No. There is a protocol that we've been using for rearing out, and Alphus rufus in Michigan State and Hannah, where we collect, let's say 100 grams, maybe not even that much, but 100 grams of ripe or overripe fruit from a couple places, let's say 5 plants, and we're using one of those deli cups. On the bottom, we're going to put some sort of sponge material, a small piece of sponge, and then a wire mesh over the top of it, and putting the fruit on top of the wire mesh. As the fruit breaks down, it leaks and stuff, and that liquid is collected by the sponge. On the top, we have a cover on the cup. So we used one of these 32-ounce cups, and then we have small breathing holes in the top of the cup, but not large enough for the flies to get out. Inside the cup, we're actually sticking one of those yellow sticky cards that when the flies pupate and emerge out of that fruit, if they do, they fly around a bit and then get stuck on the yellow cards, and we can pull the cards out periodically and look at them and identify the species, at least identify whether they're spotted-winged Drosophila. Now, would they come out in a mesh cage in the field? It's very possible you could collect them that way as well, as long as got air circulation stuff. This problem of the ripe fruit starting to decay and fall apart could be a problem, and you need some air circulation in there, etc. So I haven't tried that rearing in the field, but my guess is we could figure out a way to do that. It's just more convenient for us to bring it in the lab and put them in either on a lab bench, or we'll use some sort of screen house to rear them out and be able to check them on a regular basis. Okay, I'm going to move on. Farouk said, I just put a comment from Long Island that no spotted-winged Drosophila emerged from rearing damaged grapes last year. However, traps in the same field did capture some spotted-winged Drosophila adults. So that's something that I think Farouk will hope to do this year, as we will also up in Finger Lakes, is do some rearing out of grapes just to see when and if they become a problem in terms of larval infestation. Gilbert says, is there any indication that this pest affects the reproduction of wild host plants or animal birds that eat vulnerable wild fruits? So far, it sounds like no, but I wanted to check. Yeah, so in terms of affecting their reproductive success, no evidence that I know of that the fruit is damaged to the extent that not enough viable seeds are produced, etc. I suppose that's a risk, although I agree, I doubt that's going to be a huge problem. Whether it'll affect dispersal, my guess is this fly will be, if birds get it, they might get consumed. I'm assuming that the flies would die if they got eaten, but if they were dropped by foraging birds and things like that, it would probably not bother them. I don't think, I think it'd just be a little extra protein in terms of the fruits consuming it. But yeah, I would guess no in terms of a big impact on the success of these native plants, although I guess I wouldn't completely rule it out. It's an interesting question. Bernie Smith asks, hate to spray when fruit is ripe. What should suggest for organic sprays? Yeah, boy, I guess for organic sprays right now, unless we can come up with some of these attract and kill stations and things like that can work, and I really want to test those, and I think Rich would agree with me. We want to test some of these ideas. Chemical control. Now, we do have what is a quite effective material for chemical control, but it's expensive. So let's suppose you're doing fall raspberries, you're sort of gotten into harvest, and you find that you're seeing some spotted-wing Drosophila, and Trust is a good material that could be put on. I forget the label restrictions, though. There is some restrictions on how much you can use, so that becomes somewhat of an issue, and the expense. They're very expensive. I wish, at least right now, I don't have a great alternative. I think it would help, though, to be as much as possible keeping the right fruit regularly picked and out of the field. I hope to have some of this cold storage information. I hope we get some data on that in the short term, and that might be another approach. But I guess I don't have any great answers for organic growers right now, other than Trust. And frankly, we're lucky that we have a material as good as Trust. Some of our organic products, like Paganic, for spotted wing, are not particularly good, and they're expensive, so it's kind of a double whammy. I think Trust, although expensive, is quite effective, so we at least have something we can utilize. But I sympathize. It's something that we're not used to having to do in any of the small fruits, especially the northern areas and the northeast, in terms of spraying insecticides, and particularly the organic growers have been able to get through without having to spray a lot. And this is maybe a game changer. It may not, but I think there's that risk. Eric asks, given that we will certainly be inadvertently breeding Drosophila suzukii for resistance to organophosphates, spinosauриds, pyrethins, is there any prospect that, given its similarity to a well-studied model organism, that we could be deliberately breed for traits like smaller ovipositor, life cycle timing, or chemical cue preferences? Yes and no. Indeed, they are easy to rear, pretty easy to rear in the lab. They can get contaminated with more hardy species, but you can breed them. You could do genetics on it, and you're right. We know the genome of Drosophila melanogaster, and that might provide us insights. You know, I don't know about, I can't imagine something like smaller ovipositor, although you imagine you could breed something like that potentially through radiation and all sorts of things. Once you released it, I can't imagine that fly would be successful and replace the other one. You raised some really interesting questions about genetic mechanisms for introducing deleterious traits into populations. And there is a lot of work going on, let's say, for malaria mosquitoes. And yes, long term, that would be a pretty interesting area to pursue. I think it's a really interesting idea of using some sort of positive feedback systems that they've worked out in terms of genetic models to do that under population genetics field conditions and drive in a deleterious trait into a population like of an invasive species. I mean, there's some risks associated with that, but I think it's kind of an interesting and exciting area of pursuit. So nothing short term, though, I see there. However, knowing the genetics of it may provide us other avenues, such as using something called RNAi technology, identifying protein products that are important for the fruit fly to survive, and then interfering with that through either getting into the plant or spraying it on, and using that to in a targeted way kill this insect. And I think there's some real potential there, and having knowledge of the genetics of our melanogaster is going to promote that. And I know a number of people are looking at that. So that's some pretty interesting ideas.
Okay, if we drop down to Bernie Smith, he's asking then, when would you suggest spraying for a blueberry farm between July 1 and September 15?
Well, it maybe depend partly where that blueberry farm is, and I guess I would suggest monitoring starting in July 1st. And I guess I would be happier if you were rigorous about the traps, maybe changing the traps more than once a week, looking for adults, and then spraying when you actually detect the adults. If you're very conservative and very worried, yeah, I mean, I guess I don't have enough experience to know. My guess, based on what we've seen this year, and again, depending on where you're at, we really didn't start seeing things spiking until September. So I guess I wouldn't just jump on starting sprays until late August kind of thing. I'm hopeful, at least for this year, we're going to be safe. But I'd also say let's pay attention to our extension messages coming out for your region and more locally. We'll have these websites and we'll have email alerts coming out. And also take advantage of that. So if you have a neighbor that's starting to catch them or someone not too far away, that can also be some information that you can use to go into a decision like that. I hope that helps somewhat.
Dropping down, Richard Cowles has a comment.
Okay.
He's saying 3/4 to 2 pounds of sugar per 100 gallons will greatly enhance the activity of spinosyn-based insecticides. Thank you, Richard.
And I don't know if, Richard, you're still with us and can comment. I believe that's based on a lot of your lab work. I don't know if you've had a chance to try that under the field conditions, but I know that you have had pretty striking results in the lab. And it's like I said, this I guess we have plans to try this out under field conditions this year.
And Kay Gilbert is again talking about storm fronts. The flies literally get blown up here, and Richard comments down below, the best information we have is from New Hampshire. Spotted-winged Drosophila did not arrive until after Tropical Storm Irene. Okay, on to Jonathan Bishop. Following up now that they are here, should we be worried about earlier season crops since we've already observed them?
Well, yeah, I think there is a slight trend towards these flies showing up a little earlier each year in areas, let's say Michigan, North Carolina, where they've had them for a couple of years. And as I mentioned earlier, we have now some evidence of at least a few maggots in June-bearing strawberries at the end of the season in a field that was, not picked particularly clean. So yeah, I think we don't, let's say summer raspberries or earlier maturing blueberries. I do think we need to be paying attention to that. My best guess is we're, at least for the time being, it's the late season stuff that's the most vulnerable. But yeah, absolutely, unless there's some crazy thing and that a hard winter next winter just wipes them all out in the Northeast, I do think this is something we're going to have to live with and figure out how to deal with. We shall see, but yeah, that's my best guess, is that this is something that's going to be here to stay.
Okay, and okay. Richard responded to your inquiry saying he's just used it in the lab, but he had great data.
Yep.
And while we're waiting to see if there are any other questions, the results of the polls are there. I just wanted to point out to people, of the 16 people who responded, 7 have had spotted-winged Drosophila in their planting. And if you look down below, there was one detection in March, but the others began in July, and then the highest number in August, and then also in September.
Interesting. No, that's nice. Interesting.
And Farouk said, just to clarify the poll data, we see spotted-winged Drosophila in traps in the last week of March. That was this year, 2012. Okay. Any other questions or comments? While we're finishing, I just want to let you know I'll be sending you all an email with a link to the recorded version, so you'll be able to listen to it again later if you'd like to. I'll also include the links to the webinar, to the resources that Dr. Loeb gave us in that slide just at the end. And thank you so much for joining us. We'll be here a couple more minutes to see if there are any more questions. Thank you, Greg, for your presentation. It was very good.
Welcome. Thanks for all the comments and questions, everyone. That was great. I think we had a good exchange and plenty to do. And thank you, Kathy, for getting this organized. That was well done. We had to fight a little bit this morning, but not you and me, but fight with our equipment. But it worked out fine.
I'm really glad. I think it worked out very well, and you're welcome to keep the headset.
Oh, well, wow, nice. A present. Thank you.
Okay, there doesn't appear to be any other burning questions. We'll take all your suggestions into consideration, and we thank you for being with us this afternoon, and have a great weekend.
All right. See you, everyone. Bye. Thanks, Kathy. See ya.
Bye. Thanks.